we came to know the 7 contrasting character where Mendel searched in the pea plant. Okay, so now let's see the series of experiments performed by Mendel to prove the inheritance process. Okay, so to understand the experiments very well, there are few basic things you must understand before entering to that. Okay, so while I'll be explaining about the experiment itself, we'll talk about the basic things as well. Okay, so now we are going to see how the single gene which is responsible for a character is inherited. That means we are going to study about monohybrid cross. Okay, so what do you mean by monohybrid cross? I'll tell you later. Okay, so we'll see what experiment he did in the monohybrid cross first, and later on we'll talk about the basic things. Okay, so for his experiment, you know that he took pea plant. So he took pea plants, contrasting charactered pea plants. Okay, so one pea plant was tall, understood? Another pea plant was dwarf. That means it was short. Okay, so he has taken 2 different pea plants, understood? So 2 different pea plants means those 2 pea plants are differing in a character called as height. So one is taller, another one is dwarf. Okay, these are the parents actually. And now the genes responsible for the tall character is capital T. Okay, genes responsible for the dwarf character is small t and small t. Now we will get the question that what is this t and the small t? Okay, see, if we are expressing any characters means, that means if we are expressing any characters of our parents, that is because we have got genes from our parents. So those genes are very much important for the expression of the character. Understood? See, if the pea plant is tall, that means to say it has the genes for the tall character. So we have represented those genes in the form of alphabets. That's all. Okay. If another pea plant is dwarf, that is if it is having short height, the genes responsible for the dwarfness in the pea plants is small t and small t. You might may ask you a question that why we have representing the tall characters in the capital and the dwarf character in the small letter. Why can't we do it vice versa? It is highly impossible. I'll tell you later why it is so. Okay. So one more doubt you might be having, why we are writing the 2 different alphabets here. That means why we are using 2 alphabets instead of using only single. Why? Because see, somatic cells are diploid in nature. Means to say the body cells are always diploid in nature. So this plant is diploid, this plant is diploid. So 2n means you have to write the 2 genes here. Clear to you? Yes. Next, so when these 2 different parents are undergoing fertilization after gametogenesis process, understood? So next is we are going to write gametes. So here the capital T, see, I'll write here capital T means they are genes. Okay, so always genes occurs in pairs only. Understood? So among these genes, the first part and the second part is called as alleles. So when the alleles are together, we call them as genes. So this in the genes, the single unit is called as allele. Understood? So how many alleles we have then? 2 alleles. Correct? Yes. So in the gametes, you know that the process of meiosis is going to take place. Okay. So when meiosis takes place, it is a reductional division. So when the 2 T's are there here, after reductional division, you'll get only single T. That means it is a gene. Okay. So here we get small t. While writing the gametes, please make sure that you have to write those gametes in the circle itself. Okay, but if you don't write the gametes inside the circle, or if you just represent them like this, it appears as allele. Yes or no? Because the single unit of gene is called as allele. So you have to always represent the gametes inside the circle. Understood? So after gametogenesis, what happens? Fertilization has to take place. Correct? So after fertilization, the diploid zygote is formed. So diploid zygote means diploid. 2 alleles you have to write. One is capital T, another one is small t. So this is the first generation. So it is called as F1 hybrid, or you can also write it as F1 generation. And F1 stands for first filial generation. Understood? Yes. So here we have one capital T, another one is small t. You know that capital T express which character? Tall character. And the small t express dwarf character. Now, the half spring is expressing which character? Whether the tall character or the dwarf character? It always express the tall character. Why? Because, see, already I told you that why I have written the capital T here for the tallness and the small t here for the dwarfness. While we were discussing about the 7 contrasting characters, that is flower color, position of the flower, pod color, pod shape, seed color, seed shape, we have understood that which are all the dominant characters and which are all the recessive characters. Okay, so while we were discussing about the height of a plant, there I told you height means they may be tall, they may be dwarf. Among these 2 characters, tall is the dominant character. Only the dominant character will be expressed in the half spring. Why? Because always dominant gene expresses its character. Understood? So, here we have 2 different alleles. One is for tall, another one is for dwarf. So, which is more dominant here? The tall character is dominant. Okay. So, since it is dominant, I have written in the capital letter. Okay. So, the one which is recessive should be always represented in the small letter. Clear to you? So now the half spring is having 2 different alleles. One is capital T, another one is a small t. So here the half spring always expresses the tall character. But see here I have written capital T. It is for tall character. But here also I have written capital T and small t. It is showing the tall character. Then is there any difference between these capital T and capital T small t? Of course there is a difference. Both are expressing the tall character, but the genes are different. See here, in the capital T and the capital T, the 2 alleles are similar. Yes or no? So such condition is called as homozygous condition. Understood? One is here capital T and small t have written, but still it is expressing the tall character. Correct? See, it is two different alleles. So it is heterozygous condition. Very easy. Okay. So while you are writing parents here like tall crossed with the dwarf, you have to mention whether it is homozygous or heterozygous. Okay. So this is now the homozygous tall. And this one is homozygous dwarf. Yes or no? But here it is two different alleles, so it is heterozygous tall. This is about the first generation. Next step is there. That means also he continued the experiment. What he did is he allowed this F1 hybrid to undergo self-pollination. Why? Because pea plant is a bisexual plant. It has both the sex organs. Understood? For that reason, allowed it for self-pollination. Okay. So here we can write it as selfing. Selfing means self-pollination. So what is the F1 hybrid here? Capital T, small t. Okay. It is undergoing self-pollination. So we can write again capital T, small t. Understood? Okay. Next is again these acts as parents now. So they have to release the gametes. Since it was a homozygous condition, that means two alleles were same, they released only one gamete. But here there are two different alleles. So they are going to release two different gametes. One is capital T, you have to write it in a circle. Another one is small t, write it in a circle. Capital T, another one is small t. Understood? So these are the gametes. Next is after release of gametes they have to undergo fertilization. Yes. Then after fertilization diploid zygote is formed. That second generation individual is called as F2 hybrid. Okay. See here when you are writing the F2 hybrids, you have to represent them in a specialized box which is called as a Punnett square or it is called as checkerboard. We'll see that. So how to write this checkerboard? First is, in the first box you have to write the male and female symbols. Okay, because it is a bisexual flower which is having both the sex organs. And again here you are going to write the gametes, that's all. So one is capital T, another one is small t. So from this capital T, small t. Clear? So capital T, you are going to get this capital T, small t, capital T, small t, small t, small t. Again see here capital T is there, small t is there. You can ask me that why I have written capital T first, why not small t? Because dominant gene must be written first. That's all. Okay. So again, it is a tall plant. It is also a tall plant. It is also a tall plant. It is capital T, small t is a dwarf plant. Correct? So we have got 4 different offsprings or individuals in the second generation, that is in the F2 generation, nothing but a second filial generation. So among those 4 individuals, 3 of them are tall and 1 is dwarf. Okay, so next is you are going to represent phenotypic ratio and you are going to represent genotypic ratio. I'll tell you what it is. Genotypic and phenotypic ratio. Phenotypic ratio means it is a morphological ratio. Morphologically means the character which you can see through your naked eye. Okay, just you see this 4 individuals. Okay, how many tall plants we have and how many dwarf plants we have? Tall, tall, tall. 3 tall plants are there and 1 is dwarf plant. Correct. So the ratio is 3:1. Okay. So 3 means how many? The tall plants are there. So 1 means it is dwarf. This is called the phenotypic ratio. How we are going to write the genotypic ratio then? Okay, so genotypic ratio is nothing but, so in the phenotypic ratio you have just written how many tall plants are there and how many dwarf plants are there. But in the genotype you are going to mention whether those tall plants where there are 3, right? So whether they are homozygous or they are heterozygous in condition. See here, capital T, it is homozygous. Capital T, small t, it is heterozygous. Capital T, small t, heterozygous. Small t, it is homozygous. So here you are going to mention the ratio of homozygosity and heterozygosity. Okay, so among these 3 tall plants, so 1 is homozygous tall, 2 are heterozygous tall and 1 is homozygous dwarf plant. So this entire part is the experiment performed by Mendel. So this series of experiment you have to mention in this pattern only. While writing parents, you have to mention whether they are homozygous or heterozygous. Of course, in the monohybrid cross, they are always homozygous. Understood? Next, after writing parents, you are going to mention about their gametes. So in the homozygous condition, always a single gamete will be released, because homozygous means 2 genes or 2 alleles are similar. After gametogenesis, fertilization has to take place, which is going to form a diploid zygote, which is nothing but the F1 hybrid. Okay, so this F1 hybrid which is obtained here is tall in nature. So if it is tall, it is not homozygous, it is completely heterozygous condition. Okay, it is expressing the tall character only because it is the dominant character. Tall character is the dominant character. Understood? Next, after getting F1 hybrid, we allow the plant for the self-pollination. So self-pollination means you have to write selfing. Okay, so capital T, small t, capital T, small t. You are going to write the same genes for this also. Why? Because it is a selfing process. And here heterozygous tall is crossed with the heterozygous tall only. Understood? So gametes will be 2 different types because it is a heterozygous condition. When there are 2 different alleles, 2 different genes has to be produced. Okay? And next, you know that in the second generation, the half springs should be represented in the checkerboard or in the Punnett square. So this box what you are seeing is nothing but the checkerboard or the Punnett square. Okay. So in the first box, they are going to represent both male and as well as female. So next, followed by the gametes released. Okay. So capital T, it is homozygous tall. Capital T, small t, heterozygous tall. Again, capital T, small t is heterozygous tall. And small t is homozygous dwarf. Understood? So we have got how many individuals in the F2 generation? We have got 4 generations. Among those 4 generations, 2 individuals are expressing their parental characters. Exactly the parental characters. See, capital T, parent. Among those 4 individuals, 2 of them are expressing their parental characters. Exactly the parental characters. See, capital T, small t, parent is having capital T, small t. Understood? At the end of this, you are going to write the phenotypic ratio and the genotypic ratio. So, phenotypic ratio means you have to write how many of them are tall and how many of them are dwarf. Just the morphology are going to represent, that's all. See, we have 4 individuals, among which 3 are tall plants and 1 is dwarf plant. That's all about phenotypic ratio. Coming to genotypic ratio, okay, you are going to represent whether the tall plant is homozygous or it is heterozygous. Okay, so again, among 4 individuals or among the 3 tall plants, 1 is homozygous tall, you are going to write 1 ratio, and 2 are heterozygous tall, you are going to write 2. 1 is homozygous dwarf. So this is how you have to write the genotypic ratio. One more thing, while you are writing or representing the F2 generation in the Punnett square, you have to represent the condition of those individuals, that is these offsprings, in the Punnett square itself. That means, see here, capital T, that is homozygous tall. You have to write that homozygous tall in this box only. Here, heterozygous tall, you have to write here. So, heterozygous tall, again within this box. Small t and small t, homozygous dwarf. You have to write the homozygous dwarf inside this. Okay, and also, when you are representing the genotypic ratio like 1:2:1, how easily you can write is, what is 1? Homozygous tall. What is 2? Heterozygous tall. 2 are there. What is 1? Homozygous dwarf. Very easy. Okay, so this is about the monohybrid cross or the inheritance of a single gene. Inheritance means transfer of a single gene. How the single gene is getting transferred into offsprings.